Hi everyone. So this is welcome to my channel Pure Biology, and today's topic is the enhancer or the silencer. You just actually think about what is the meaning of the enhancer and the silencer. Enhancer meaning that enhance the transcription process, because enhancer and or silencer that involved in the transcription process. So just I am saying the topic transcription related to enhancer and silencer in that way. So enhancer means that initiate the transcription process, or you can say that it tries to continue the transcription process, and silencer, what is the role of the silencer? That tries to stop the transcription process. Okay, this is one kind of protein. So what is saying that many genes in mammalian cells that are controlled by the regulatory sequences. There are some regulatory sequences that control the mammalian genes, okay, and that located were farther away from the transcription sites. Actually, the transcription process initiate from the place the presence of enhancers or the some regulatory sequences, they remain just farther away from that site, okay, and these sequences may be enhancers or the silencers. So what is saying that these enhancers or the silencers, these are one kind of regulatory sequences and that located farther away from the transcription sites. That means from the distant region from where? From the actual transcription site or transcription region. Okay. These enhancers are the transcription control elements, and they serve to increase the basal level of the transcription, which is initiated through the core promoter elements. So its role is that I already told that enhancer means that enhance the process, and silencer means that tries to stop the process. So enhancers actually this increase the basal level of the transcription, which is initiated through the core promoter. Actually, which is already initiated by the other factors, that is the promoter and the transcription factors or the RNA pol. So already have started the initiation process, but this is the initiation process is maybe increased or decreased by this enhancer or silencer. Actually, you can say that enhancer or silencer that act like the catalyst. Okay. So either it enhance or it tries to silent. Okay. Now this typical enhancer, that is its BP means it is one kind of protein. So its length is 100 to 300 BP and essentially consists of an array of binding sites for DNA binding transcription factors. So it consists of binding sites for which, because this binding sites is related to the binding of the DNA, okay, for the DNA binding transcription factors. That means the binding sites consisting of this enhancer for which this place is the site for binding of the DNA and obviously some transcription factors. Next is that this is one kind of enhancer, SV40, and SV40 is seen in some eukaryotes or viruses obviously. So some characteristics or the features have taken from this SV40 and that is explained here. So first one is that this is cis-acting DNA sequence. Cis-acting. There are two sites of transcription layer. One is the cis layer, cis site, another is the trans site. So enhancer always tries to bind with the cis site, and that strongly activates the transcription of a linked transcription unit from its correct initiation site. So this activates the transcription process of a linked transcription unit from its correct initiation site. So actual initiation site is the region on which the enhancer play their roles, or they tries to increase the continuation process actual initiation site, not the false region. Okay, it first identify or recognize the initiation site, then acts on that site. The second is transcription independently of its orientation. Orientation means from where the transcription process is initiated, that is called the orientation place. So on which the enhancer that acts. It is able to exert its function over large distances of more than 1000 BP from a position either upstream or downstream of the initiation site. So its capability is about more than 1000 BP. That means it's a function over large distances. That means it can act from the large distance also, about from how much large? That is the 1000 BP. Okay, this from this area or the limiting site, you can say that the 1000 BP is the limiting site. If that enhancer is there about the 1000 BP away from the initiation site, so it's the enhancer can play their role. And if it is downstream or the upstream. So enhancer stimulates the transcription in a cell type specific manner, and the transcription factors bound to distant and enhancer can thus work by the same mechanism as those bound adjacent to promoter. So activity of the enhancer and the promoter just like same, not completely same but likely same. So so it is there is very little differences is seen between the promoter and the enhancers. The elements analogous to the enhancer that called the upstream activation sequence are found in yeast. This is the sequence, one kind of sequence, and this sequence is related to the eukaryotes or the yeast. Okay, yeast, and here the enhancer is related with this sequence. And this is found in the yeast. And at variable distances upstream of the promoter but cannot function when located downstream. So what is the actual function of the enhancer? Enhancer can play their role where only in the upstream region. So if it remains more distances but obviously a limited range, but if this is present in the upstream region, so it can play their role. Okay, but if this is remained in the downstream region, in that cases their activity or their functions is not seen. And it is very specific DNA element called the insulator that is placed between an enhancer and the promoter. It prevents the enhancer from activating the promoter. So insulator is another protein. If insulator is present just between the enhancer and the promoter, then the promoter, which is recognized by the enhancer and increase their activity by the enhancer, that is totally stopped by the presence of this insulator. So in brief explanation, this is main thing. The enhancer generally increase the activity of the promoter, okay, for doing the transcription process smoothly. This is the role of the enhancer. And when it becomes silencer, when it played very negative role on the promoter, so in that cases it is known as the silencer. So this is the topic of the enhancer or the silencer, and how this enhancer and silencer involved in the transcription process. So it is explained here briefly. If this channel is very helpful to you, so please you share to your friends for getting the knowledges. And after subscription or after subscribing the channel, you just hit the bell icon for getting the notifications of all videos. Clear? Thank you.